Alright guys, welcome back to Underground Science, and in this video we'll be talking about the enzymes related to the Krebs cycle. So in the last video we talked about the compounds created in the Krebs cycle shown here, alright, and in this video we're going to be talking about the enzymes involved in catalyzing each of these reactions in the Krebs cycle. Alright, so let's just start off by listing all of them. Okay, so one thing that we have to notice here that we didn't put is that remember that when oxaloacetate and joins with another molecule that comes from the transition phase of glycolysis to make citrate. If you guys remember, that molecule is acetyl-coenzyme A. Alright, so acetyl-coenzyme A. Now, we're only drawing one acetyl-coenzyme A, but there's actually two acetyl-coenzyme A's involved, alright? That's going to be really important to not forget that there's two acetyl-coenzyme A's involved when we talk about the byproducts produced in a future video, alright? But for now, let's just focus on the enzymes, alright? And let's just say that we have one acetyl-coenzyme A coming in at a time, but we know that, just keep it in the back of the mind for a future video when we talk about the byproducts produced in the Krebs cycle, that we actually have two acetyl-coenzyme A's coming from the transition phase of glycolysis because we had two pyruvates, right? So let's go ahead and get started talking about the enzymes. So first enzyme involved in the catalyzation reaction between citrate to isocitrate is going to be called aconitase, all right? So aconitase, all right? So aconitase. All right, so this enzyme is going to react, or is going to catalyze this step, all right, between citrate to isocitrate. So citrate to isocitrate, and that enzyme is aconitase. All right, and then next enzyme that's going to be involved in the catalyzation reaction between isocitrate and alpha-ketoglutarate is isocitrate dehydrogenase. All right, so isocitrate dehydrogenase. And notice that I didn't draw any of the things that we drew in the previous video about like CO2 leaving a few areas, and I didn't draw the succinyl coenzyme A coming here because I didn't want to like make it too crowded because we're just talking about the enzymes in this video. But make sure to go and check out my video on the compounds produced in the Krebs cycle so you can see those succinyl coenzyme A groups that came in this reaction, and the carbon dioxide molecules that left here, and so on, all right? So in the previous video, I didn't mention it, but we'll see in this video, or let's just mention it right now, is that when we go from fumarate to malate, our enzyme involved here that we'll see in a moment is going to put water into this reaction. All right, so we're going to have water come here. All right, so just put that there so we don't forget about it. So isocitrate dehydrogenase is going to come here and act as a catalyst, lower the activation energy for the reaction between isocitrate to alpha-ketoglutarate. All right, next step, or the next enzyme involved from the conversion between alpha-ketoglutarate to succinyl coenzyme A is alpha-ketoglutarate dehydrogenase. So those enzymes are pretty easy to remember, all right? So alpha-ketoglutarate dehydrogenase, alpha-ketoglutarate dehydrogenase. All right, so that's that. So this enzyme is stimulating or catalyzing this reaction between alpha-ketoglutarate to succinyl coenzyme A. Now, when we go from succinyl coenzyme A to succinate, we don't use a dehydrogenase enzyme, we use a synthetase enzyme. And in particular, we use succinyl coenzyme A synthetase. All right, so succinyl, we'll go ahead and write that down right here. We use succinyl coenzyme A synthetase. All right, and again, I didn't draw it here. Let me go ahead and draw it for you guys. That we add a coenzyme A group in the reaction between alpha-ketoglutarate to succinyl coenzyme A, right? Our alpha-ketoglutarate dehydrogenase adds a coenzyme A group to it. And then right here, you can see that it becomes succinyl coenzyme A. This coenzyme A group gets added on, but then it leaves again and turns into succinate. So our coenzyme A group leaves this reaction again. All right, so it gets added on and leaves. So that's that. That's good to mention. All right, so after that, after succinyl coenzyme synthetase synthesizes or catalyzes the reaction, I should say, between succinyl coenzyme A to succinate, then we have succinate going to fumarate. Now, what enzyme is catalyzing this reaction? And actually, this enzyme, I should mention, is important, is actually bound on the cristae of the mitochondria. So let me go ahead and draw an arrow here and say where this enzyme is going to be the succinate dehydrogenase. All right, and one thing I want to mention is that I noticed in my last video when I talked about the compounds produced in the Krebs cycle, I actually drew the mitochondrial organelle, but it was getting too crowded. So I just drew it right here in the open, but know that all of this is happening in the mitochondrial matrix, all right? The whole Krebs cycle cellular respiration process is happening in the mitochondrial matrix of the mitochondrial organelle in our cells. Okay, so succinate dehydrogenase is going to be a special enzyme because not only is this enzyme different from the other enzymes in the Krebs cycle, as in the fact that it's bound to the cristae or the inner mitochondrial membrane of the mitochondrial organelle, but that it's also involved in the Krebs or in the electron transport chain. All right, I was about to say Krebs cycle. It's obviously involved in the Krebs cycle, but it's also involved in the electron transport chain that we'll see in a future video. So let's just say for now that it's bound to the cristae of the mitochondria. All right, so succinate dehydrogenase comes here and it catalyzes the reaction between succinate to fumarate. All right, so then next, what enzyme catalyzes the reaction between fumarate to malate? Well, this enzyme is going to be called fumarase. All right, so fumarase. Fumarase. All right, and fumarase, again, like I said, adds a water molecule in this reaction and catalyzes the reaction also between fumarate to malate. Now, the enzyme that catalyzes reaction from malate to oxaloacetate is called malate dehydrogenase. All right, malate dehydrogenase. All right, and last but not least, guys, the enzyme that catalyzes the reaction between oxaloacetate forming citrate by combining acetyl coenzyme A is going to be called citrate synthase because we're synthesizing citrate. All right, so it's going to be our citrate synthase. All right, so we've finished our enzymes of the Krebs cycle video. Right, so we have citrate synthase, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. Then we have succinyl coenzyme A synthetase. Then we have succinate dehydrogenase that we highlighted specially because it's bound to the cristae and involved in the electron transport chain. Right, so let me just go and write that down also, that it's involved in the electron transport chain, but we'll look at that when we talk about the electron transport chain. So this is also an enzyme in the electron transport chain. All right. Okay, so continuing, we have our fumarase afterwards, then our malate dehydrogenase, and back to our citrate synthase and aconitase, and the cycle continues. All right, and just know that in our next video, when we talk about byproducts produced, know that we have two acetyl coenzyme A's, okay? Because then we'll know that for each acetyl coenzyme A, the Krebs cycle turns once. So that means every byproduct that we produce is going to be two of each. So you know how we said that in the previous video, we said that when we go from isocitrate to alpha-ketoglutarate, we lose a carbon dioxide molecule. We don't lose one here. We lose two carbon dioxide molecules because we were talking about one CO2 molecule because we were talking about one acetyl coenzyme A molecule, but there's two. So we don't lose one carbon dioxide molecule here, we lose two. Same thing goes for alpha-ketoglutarate to succinyl coenzyme A. We don't lose one CO2 molecule, we lose two. All right, so we lose 4 CO2 molecules in the Krebs cycle. And small things like that, like right here, right here, one coenzyme A doesn't come in. Well, one coenzyme A comes in for every one acetyl coenzyme A that we started off with. But this Krebs cycle has to turn twice to compensate for the 2 acetyl coenzyme A's. So here, 2 coenzyme A's will come in, not just one, because we have 2 acetyl coenzyme A's in the beginning joining with the oxaloacetate, right here. So just things like that right here, 2 coenzyme A's leave, not 1, because we have 2 acetyl coenzyme A's in the beginning and we join with oxaloacetate. And then again over here, I know it's kind of repetitive, but we have to focus on this, that there's not 1 water molecule that's getting put in. Fumarase puts in 2 water molecules because we have 2 acetyl coenzyme A's. Our Krebs cycle makes 2 turns, 1 turn for each acetyl coenzyme A. Alright, so I hope that this video made sense to you guys. In my future videos, I'll be talking about more cool topics about biochem, physics, and chem. And in particular, I'll be making a video soon about the byproducts involved and substrate level phosphorylation involved in the Krebs cycle. So hopefully you guys tune into that. Make sure to hit that notification bell, like and subscribe, and see you guys later, and stay safe.